Hello, everyone. So it's almost all. Welcome back. I hope you had a good lunch and break with your some fun with your artificial sensory neuron. It was quite easy. Uh, I think, sorry, I think I misunderstood the question that I'd been asked before by Simona, I guess, about the assignment. You were referring to the three sections, right? Not to the three question of the first task. Okay, yes. No, of course, like the other two, you still needed to be to understand what was behind that. So you could not have possibly known what to do or what to pay attention to. Guys, it's always the same few people that switch on their camera. Can we try to amp up the numbers here? Good. It gets a little bit better and better. Hi, Kevin. I see Kevin. All right. So there is not much really to discuss. There is a lot, like already the features of the receptive field that you have discovered through your exercise were being mentioned during the lecture. So if you have discovered what they were responding to, perfect. Otherwise, it's in the slides. So check it out. We are going to go with the next part in which we're going to go deeper and deeper into the brain, into the areas that take care of not only take snapshot of the sensory world that is around us, but to attach meaning to it. And this is particularly close to us in the Donato Lab because this is literally what we work on. So if you are curious about this or about our experiments or what this drives our questions, just ask things away. Okay, first of all, is there any question that might have come during this break that you want to answer before we move on to talk about sensory coding or coding in higher cognitive areas? I see that there is no question on Slido, so anybody has a face-to-face question?
When do we have to hand in all exercises?
Yeah, by Sunday, Saturday, Sunday. It's not a hard deadline for Friday. Again, try to understand what the concepts are about and how they can prepare you to play around a little bit, and that would be perfect. Okay, so there seems to be no further question. We move to the brain. All right, yes, we are ready. So this morning we have seen a few examples of cells responding to stimuli that come in the sensory world, meaning that we saw that in the primary visual cortex, we have neurons that are able to extract information about or to detect some feature of the sensory stimulus. In this case, the orientation of bars, but also we have talked about the movement, contrast, color, and so on and so forth, even if we haven't seen the specific examples. We've also talked since yesterday, and I think you have already heard it also last week during the blog course, but in your course in general, that there is a progression of information, or if you want a transfer of information for primary areas that receive direct information from the thalamus to other areas which prevalently start to get information from other cortical areas. So we go from V1 to V2, V4, then from there directly to IT, and then from IT we enter into the loop that then brings information down to the hippocampus. So one of the questions that you might come to think of is if we stick an electrode in different areas of these and we present the same stimulus that we presented in V1, what would tuning curve look like? And the answer is that we would find more and more complex feature to which the neurons respond to as we go higher in the cortical hierarchy. So if we want to take a very simplified and schematized example, let's suppose that we are in New York City when we are going to be able to travel again, and we're looking at one of my favorite building in the city, which is the Flatiron building. We are going to be sticking our electrodes in our V1. We would see that neuron starts to respond at features, for example, the orientation of some edges between the different elements of the building. In V2, they would start to combine these edges in ways to get a complex type of shapes, like for example, corners. And then you would start to get the outlines to detect basically the individual elements like the windows, this part of the building that signifies an increase of another floor or the whole outline, until we arrive in area IT, which is the one that we already saw before. And we see that the neuron there might responsively present one specific type of sensory stimulus, like one specific house, but not another one. So full object represented in the activity of this neuron. Now, this is just part of what the brain does. Yes, right? We talked about Monday from the fact that about the fact that the brain is not only able to create an internal representation, if you want, of the sensory world, to create a mental picture of the visual scene. We have defined this process perception, but is able to attach meaning to that by modifying the signal that basically is represented into the sensory areas. So touch meaning, like for example, remembering that placing the Flatiron building at a specific location in the map of Manhattan, or start to think that the Flatiron building is one of the instances of objects that we can define together as Manhattan building, like we have the Chrysler building, we have the other building that I recognize, or it might just elicit what we have seen with the tennis ball and Federer, a fact that we are associating a positive valence to it, meaning the fact that we think, okay, this is a beautiful site to look at. This is what we have defined before as cognitive processes, right? Being able to extract general information from the object that we have in the single word, attach valence that is associated to our previous experience, or able to place them in a spatial map of the environment in which we move in. But where does this happen in the brain? And to understand what cognition happens in the brain, where cognition happens in the brain, we need to understand what happens in these areas of the brain, right? The ones that are located quite far away from the sensory areas. One of these areas, for example, is the hippocampus, as we saw before. And I want to report here a quick experiment. It was very influential both for the field of neuroscience and for the field of general knowledge in general. So in 2005, a group of people in LA, they were guided from Rodrigo Quiroga, but it was basically one team with Christoph Koch that some of you might actually know because he's quite famous in the field of consciousness, and with Isaac Fried, who is a surgeon. So there were a series of patients that had epilepsy. And one common thing that when people with epilepsies go to be cured have to go through is the fact that they start to place some electrodes in the area of the brain to identify which area is actually the source of the epileptic seizures. And of course, once you put the electrode, you basically listen to the activity of this neuron to start to understand when the synchronized activity happens and identify the seizure. But this also gives people, and surgeons especially, a favorable window in which they can, while the patients are basically waiting in the hospital for the seizure to happen and them to be able to record from this seizure, they can go through some easy experiment, and they can allow them to, for example, look at the activity of individual neurons while they're presented with some pictures. And that's what these patients were doing. So Rodrigo was showing them pictures, and this you see like picture number here, and recording what was the response of the individual neurons, of some individual neurons, to the presentation of these pictures. And he saw that there was few neurons that were particularly responsive to specific pictures. They would not respond to all the pictures. If you see here, out of 90 pictures, they respond maybe to 10. But to some of those pictures, they were responding very, very strongly. So if I ask you now, what do you think was represented in those pictures, by remembering things that I said this morning, what would you think would be the answer?
To which picture we pay attention?
No, there was something, they would pay attention to all the picture. And this is something that you can actually track, right? You can actually even tell them what is represented in this picture, what you see. So you know that they recognize the picture. But there were some pictures that were able to elicit a response from this neuron, So if you remember the lecture of this morning, these were the picture where you actually started, this neuron started to increase their firing rate, and some pictures that did not elicit this response. So based on that slide that I showed you today, who was in this picture? Come on, guys, it's easy.
Well, Jennifer Aniston.
Great. Who sings the song, the intro song of Friends now? Come on, it's easy. Sing the song. No, it was indeed Jennifer Aniston. So these were some of the pictures that were presented to the person. And this is basically the Jennifer Aniston neuron, which became a sensation in basically the whole world. I mean, if you Google Jennifer Aniston neuron, you see a lot of articles about it. So if you see here, there is Jennifer Aniston, which was very famous in 2005. And you see that there is a group of 7 pictures to which the neuron starts to fire very highly. So you see that before the presentation of the picture, the neuron is quite silent. After, when the picture is on, which is in between these two staple bars, the neuron increases firing rate, and then it goes back to baseline after the picture has been withdrawn. The picture does not fire when the late Kobe Bryant is shown. It doesn't. This was LA, right? So he was the most important and probably one of the best players ever in the NBA. The neuron would not fire when Julia Roberts was presented, Pamela Anderson, some people that I don't know, a snake, a spider, a deer, a dolphin, a house, the Tour Eiffel, the bridge in San Francisco, the Opera House in Sydney, you name it. So basically, the neuron was quite specifically responding to picture in which there was Jennifer Aniston. It's interesting to add something here, that is that this neuron was not responding to the Twin Tower picture. It's actually a quirk. This was done in the US in 2005. This was actually a very emotionally charged stimulus. And this was actually controlled to see maybe the neuron responds to something that the person particularly likes. So it's responding to something that elicits a strong emotional response. And this was actually the indication that the identity of the stimulus was actually more important than the emotional valence of the stimulus. Anyway, one interesting thing to notice, though, is that this neuron was not responding to seeing the face of Jennifer Aniston, because Jennifer Aniston was also in picture number 7, in picture number 6 or 67. It was just paired with the then husband, Brad Pitt. So this neuron is not responding to the face of Jennifer Aniston. That is what you would expect from an area that gets information from area IT. If you remember, I told you that area IT is able to recognize faces. So what information about Jennifer Aniston was represented in those cells? And to understand that, we need to go to another series of pictures. In this case, you find a neuron that responds to Halle Berry, which was, again, very popular at that time, and which really exemplified something extremely interesting. So the neuron was responding to the picture of Halle Berry, first 4, to a drawing of Halle Berry, to Halle Berry dressed as Catwoman. The movie had just come out. But the neuron was also responding to just the name Halle Berry written on the screen. So it's clear that this neuron is not responding to the same type of sensory stimulation all the time, right? Because otherwise a name written down is very different from a face of a person, which again is quite different from a drawing. So what they started to imply and to hypothesize is that the neuron was not coding for information that directly received from the sensory neuron. They were not locked to single sensory modalities, but they were able to integrate information that came from the sensory stimulus and from previous experience and respond to the concept of something instead of the something that was included to the neuron itself. So in this case, it was the concept of Halle Berry or the concept of Jennifer Aniston, and not the figure of Jennifer Aniston itself. Is that something that passed through, or is it the difference between the sensory stimulus and the concept of the sensory stimulus? It's something that I think Nuno was asking in one of the previous questions, right? The neuron does not respond only to the zebra, but it responds to the concept of something that has stripes on its body. And it would respond to different animals in that case. is something that basically like general features to which the neuron responds to that predict the response of the neuron. And this highlighted one of the principles of coding in high-end cortices, which is the fact that these neurons lose touch with the individual sensory modalities because they're far and farther away from the sensory responsive area of our brain. But they're able to listen to multiple sensory modalities at the same time and to integrate this sensory modality to create representation that go beyond the immediate sensory experience. Even if you stimulate the retina with different stimuli, a picture of a person, a drawing of the person, or the name of the person, the neuron will make the connection that these three things belong to the same type of concepts and then will respond to each of them, even though the way in which the sensory areas are stimulated by the three stimuli are different. This was quite important because it was one of the, first of all, because it's one of the first type of representation that goes extremely, in a way that is extremely abstract and controlled in comparison to the sensory stimulus, but also because highlighted and sort of for a certain period of time even confirmed what I've been telling you before, that is that as further away as you go from the sensory area, you should be able to put together features to abstract more and more from the immediate experience that you have and go back to the idea of having a cell that responds when you see your grandmother or when you think about your grandmother. That would be the response of the to the writing of the name of Halle Berry in that case. Now, here neurons are not tuned to a single physical stimulus of the sensory world, but create tuning curves that are abstract and that are created almost entirely by the type of computation that go on at the level of the brain. So it turns out that the hippocampus is actually a very good model to do that because it's able to listen to different and multiple sensory modalities and able to integrate information of multiple sensory modalities with information that comes from previous experience and with information that comes from the internal state of the animal in order to create those type of cognitive representation, or at least a part of it, that we have seen before. The question comes, how do you create these very complex tuning curves, which represent not something that is physically present in the sensory world, but something that integrates a lot of different variables at the same time? And how do you use this type of representation to guide your future behavior? And the hippocampus becomes our case study for answering these type of questions. We're gonna look at this in from two different aspects, which incidentally are also the way and the line of research that our lab follows. That is today, the role of the entorhinal hippocampal network in the representation of space. And tomorrow, the role of the entorhinal hippocampal network in the formation of memories of everyday experiences. So when I talk about the entorhinal hippocampal network, I've highlighted here in the human brain what I'm talking about. There is this area of the brain, which is in the medial temporal lobe, in which you find the hippocampus proper, this green area. You find the parahippocampal cortex, that are this shaded area here, the perirenal cortex, which is this area yellow here, and the entorhinal cortex, which is the area that basically like sits next by to the hippocampus. As part of this limbic system, we also have the amygdala, but we are going to talk about the amygdala briefly tomorrow, and we are not going to go in super high detail about that. We just will focusing on the connection between the hippocampus, entorhinal cortex, and mainly the entorhinal cortex. So to go through the first, so any question first of all about this? Any curiosity, something that you want to ask about before we move into the role of the hippocampus in the representation of space? Okay, so we'll go on. First of all, let me go just a moment back into trying to understand and present the research on the hippocampus from sort of an historical perspective. Navigation is something that has been extremely important for humans since the dawn of time, really. And people have tried to navigate, to find new lands, to find resources for food, but it's an extremely conserved behavior that animals use for everything concerning with their survival. They want to find food, they want to find shelter from predators, they want to find a mating partner. So navigation is really one of these fundamental behavior that spans, I would say, almost all the phyla of living being. In mammals, navigation has been thoroughly studied in the 1940s by one guy in particular, which was Edward Tolman, who was able to study the way in which rat navigated through mazes. So he would put the rat in a maze, you would put the rat to just randomly forage for food. There is a piece of food in the maze. After the rat has found the piece of food, he can go back to the home location where he started his journey, so that he can find, so that he can basically be picked up, a new trial can be started. What Tolman identified is that when a rat moves around into a box, and here you have, for example, a circular box, is able to move around and navigate through the box to try to look for these pieces of food, moves first to the right, then to the left, then arrives to the point when actually the piece of food is found, and he immediately is able to go back to the home location in a straight path. So it doesn't need to go back to trace back the step that is done, but it can directly find the shortcut that brings him directly in a very linear path from the place where he is to the home location. And you can think, okay, the rat can simply see where it started, find it in the environment, and just, go back there. The interesting thing is that if you do the same type of experiments in complete darkness, in which the rat cannot actually see the environment, he's still able to navigate the environment, to move first left, right, then move a little bit up, rest a little bit in this position here, and so on. But then when he finds food and wants to go back to the home location, he can still go with a very straight path. Which means that somehow the animal has been able to do some sort of computation which is not trivial. He has an idea or a map in his mind of where he is in the environment. First of all, a map of the environment itself, for which he can place himself into the map. That is basically understanding the famous signal, you are here, when you go into a map into some new location. And he can locate himself into this cognitive map on an instant-to-instant basis and be able to calculate displacement vectors. First I went left, then I went right, then I went left again, in order to have at every moment an idea of where in the environment the animal is, and being able to take straight path to the environment, to the home location. This is a type of computation that is known as path integration, and it is something that basically humans have used for navigating into the sea, or to go to the US by boat and stuff like that for a long period of time. Path integration and the ability to find shortcut needs an animal to be able to use previous experience to build a cognitive representation of the environment in which the animal moves, which he defined as cognitive map, to represent itself within this map, to calculate the direction of his movement, the distance travels, and his velocity, and to track and update this position in the map in agreement with its real world trajectory. These are things that are not trivial, because the animal has to basically have a precise representation of all these variables at the same time, and to use it in an instant-to-instant basis to decide where to go. So the question immediately prompted, is this real? When he put forward this cognitive map hypothesis, was quite controversial. You remember from yesterday, it was still the moment in which people thought we cannot understand the brain because we cannot look into it. But luckily, new technology developed, and especially with the advent of the ability to record from single neurons in freely moving animals, with the development of the single unit electrodes and the tetrads later on, it was able to look for the neural correlates of this phenomena. And to look for the neural correlates of this phenomena, people started to look in the area of the brain that is known as, you name it, the hippocampus. And why? I told you this morning that you're guided through to make hypotheses about the function of an area in situations that occur naturally most of the time, in which a person does not really have a part of the brain, either because it has been a stroke, either because there has been a lesion that is induced by a virus, or because people have misjudged, or surgeons have misjudged some circumstances and perform operation that they didn't know would cause consequences. This happened in the 1950s when Henry Molaison, who was a 27-year-old person, went to the doctor because he was suffering from intractable epilepsy. And the doctor thought, you know what, your epileptic seizures are located in your hippocampus. We're going to just remove the hippocampus and the area around, so that we remove the tissue and you're not going to suffer from epilepsy again, and you're going to survive. And they exactly did that. So if you see, like, patient HM, as it has been known for a long time, did not have his hippocampi anymore. The only thing that they didn't know at that time is that these would leave them permanently with permanent deficits. And these permanent deficits were in its ability to orient himself in a new location that he had never visited before, and in the ability to create new memories. This is the type of things that, if again you have seen the movie Memento that I was like reminding you yesterday, is the type of impairment that removing the hippocampus does. You cannot create new memory. So people started to look at the hippocampus to find the correlates of this cognitive map because these orientation deficits. And let me just introduce you what the hippocampus is. The hippocampus is a part, again, of the medial temporal lobe that is connected very strongly with the entorhinal cortex. The entorhinal cortex represents, if you want, the source of information. If you want the interface between the hippocampus and the rest of the cortex, it gets information from sensory modalities and from the prefrontal cortex. That is then conveyed to the hippocampus. The entorhinal cortex is divided in two regions, the medial entorhinal cortex and the lateral entorhinal cortex. They both project to an area of the hippocampus that is called dentate gyrus, which is the input, if you want, the input area of the hippocampus. The dentate gyrus projects to area CA3 of the hippocampus. The area CA3 of the hippocampus projects to area CA1 of the hippocampus. And the area CA1 of the hippocampus projects to subiculum and back to the entorhinal cortex. So this creates a loop, for which information enters into the loop through the superficial layer of the entorhinal cortex. It's propagated through the 3-synaptic loop of the hippocampus, and then it's again relayed back to the deep layer of the entorhinal cortex, from which it goes out to the rest of the brain. What I really need to understand is that the different area of the entorhinal hippocampal network might have different functions, and that people like John O'Keefe, who was really the pioneer of these studies, decided to try to understand the function of the hippocampus, and at the same time trying to find the correlates of this cognitive map by implanting microelectrodes of the hippocampus of rats, while these rats were moving freely into a box, in the same type of experiment that Tolman had hypothesized, right? So he had two intuitions. Until then, Hubel and Wiesel, that we heard about this morning, had used anesthetized animals in which we present sensory stimuli in a very controlled fashion, but in a very passive way. So the animal is anesthetized, is just made look into a screen. We present some sensory stimuli on the screen, and we record the activity of these individual neurons to that particular stimulus, which is not really telling us if the neuron responds or is important for a specific behavior of the animal, because the animal is not behaving, is under anesthesia. The intuition of John O'Keefe was instead to study to align the activity, the neural activity of the animal, so the neurons that we record, to actually the behavior of the animal itself, in a way that we can make a bi-directional, if you want, correlation, and understand if there are some specific things that the animal does which elicit a response in the neuron, and some specific responses or activity of the neuron that induce the animal to do something. The encoding and decoding problem, right? And this became really a very important way in which people study this phenomena. You can see here one example of this recording in which you have a rat implanted with a hyperdrive and tethered to a cable, as we have seen on Monday, just running along into a box. When the animal is doing something, stopping, looking ahead, looking for food, try to find these small crumbs of biscuits and so on, we can listen to the activity of its eating at this moment. We can listen to the activity of this neuron and understand if there are neurons that are active at specific moment of the animal's behavior. And this is really like the way in which now we are trying to dissect the correlates of the cognitive map. We are trying to align the neural activity of individual neuron to the behavior of the animal. So studying the behavior of the animal becomes extremely important because it becomes the source of these stimuli that we try to correlate with the neural activity of individual neurons. Is that correct? This was a very important paradigm shift in neuroscience. And it's very nicely explained in a book that is called The Hippocampus as a Cognitive Map, which is basically one of the pillars of our fields. Anyway, first of all, let's go to Google in this case. So what would you expect to find in an area that takes care of creating a map and basically supporting navigation? If we have an intuition of how Google Maps works, we expect to find something that answers to the question, where do you want to go? a certain form of representation of different places. Like we have here, like location and tags for Italy, France, Belgium, Germany, Czechia, and so on. We would need to find a place to encode the information where we are and how far the places where we want to go is in order to plan our journey. We would need to find a way to understand where we are heading. So something that acts like a compass in order to navigate the map where we actually are. And something that allows us also to understand on a moment-to-moment basis that there are obstacles on our way that we cannot really superate, right? For example, if you want to cross, if you're in Trondheim and you want to cross from one to the other part of the River Nidalva, you need to go through the bridge. Otherwise, you end up in the river, right? So there are all these different type of information are needed for us to navigate a map. So do we have this machine in our brain? The thing that John O'Keefe did in the 70s was actually quite ingenious. He first lowered electrodes in area CA1 of the hippocampus, but the same type of things you see if you lower your electrodes in area CA3 or in the dentate gyrus. And he recorded the activity of individual neurons while the mouse was moving. And I'm going to show you now a movie of this in which you're going to see something interesting. So this is the rat moving. The gray line here is the path of the animal. There will be a red spike, a red dot appearing every time this individual neuron we are listening to is active. And here you will see basically like the 4 different waveforms that are picked by the different channel of the tetrads that is used to record this neuron, so that you can connect this information with what we heard on Monday and understand that this is indeed always the same neuron. So let me just play the animation for you. This is of course speed up. This is usually 20 minutes of recording. You start to see something interesting, that this neuron is not active where the animal is in every position of the environment, right? It seems to be quite specific to be active in one area of the environment and active most of the time that the rat cross to this particular location. So if you, in the end, use the information that you've got from your spike and do a density plot to understand how many spikes there are and were overlaid when the animal was in a specific location, so you see that the neuron is more or less silent everywhere else in the room, apart from when the animal is in this specific location. And this is what he called the place cells, and what we started to talk and study in the hippocampus as one of the type of neural representation that might be at the basis of what Tolman had said was the cognitive map. So the place cells are neurons that are able to increase their activity when a mouse or a rat passes through a specific location of the environment. I want to highlight one notion here. It does not matter where the animal is coming from or where the animal is going. The place cell fires always at that specific location. That is called the place field. This place cells here is always active at this corner. It doesn't matter if the animal arrives from this direction or it's moving into the other direction. The same things for these place cells and the same thing for these place cells. This highlights one of the features that I told you before. In the different path, the animal is experiencing different sensory cues. In this case, for example, when he moves toward the wall, he's going to be seeing the wall and this part of the visual field. When he's moving away from the corner, he's looking at this part of the visual field. It doesn't matter if the animal is in that position, that cell has a very high probability of being active.
Sorry, question?
Yeah.
What would happen if you change the system during the experiment? For example, when you change the system or the parameters, so for example, you can move the walls and make the place bigger. So you start with this condition, and after 10 or 5 minutes, you increase the area a little bit for like 5 or 10 centimeters, so that you change the parameter.
Yeah, so there are 3 things that can happen. In one case, the animal would say, okay, now this is a completely different box. I will not be active anymore in this location. I will go silent, or I will move my fighting field in another place. This is what we call remapping. It's the way in which you basically create different representation for different rooms. Another case is that the cell will follow and say, you know, my place field is at this corner. I will follow the corner wherever the corner goes. So we will just move with your manipulation. The third thing that can happen is that this becomes stretched, so that you get one map and you try to enlarge it more and more. This is very specific for the representation that you see in the hippocampus. I'm going to show you a representation in another brain area, which does not respond in this way. It's a universal map that in that case, if you move, it will just fire, it'll increase the number of firing fields that it has. So hold your thought for a moment. Now, the interesting things come. I told you about remapping, right? That you have different firing location in different rooms. For example, imagine that this is always the same cells and these are 3 different rooms. The cell would fire in different locations in each room to signal that this is actually a different room. If you instantaneously will switch from one position to the other, if you teleport the animal from this room to this room, the play cells will almost instantaneously switch as well, but there will be a moment in time, which is very short, it's 120 milliseconds, in which the 2 types of representation will compete with each other to say you are in room 1 or you are in room 2. And the winner will basically take over and the other one instead will go silent. These are experiments that have been done. It's something that I found particularly cool, and it really tells you the flexibility and the importance of having some sort of representation that is very cognitive and away from sensory stimuli. Okay, the other information that I wanted you to have from this slide is that as the animal moves along an environment, in this case you have a linear track, you have different population of neurons that are active at different locations of the track, so that in the end, when we this afternoon are going to talk about population coding, you can create a map of the whole space that you have done, and you don't have just the representation of an individual segment of the corridor. Few years after, John O'Keefe and Dostrovsky recorded from the hippocampus CA1, someone else did something similar, but just looking at the subiculum. So they lowered the tetrodes in that case, yes, it was tetrodes already. So they lowered the tetrodes in the subiculum, and they started to record cells with the same during the same behavior that John O'Keefe had done. And what they saw was something striking. So they basically saw that there was not real place specificity, right? The cells was firing more or less in all the box. But if you were looking at individual trajectories of the animal, this cell was firing always while the animal was pointing its head to a specific direction. So for example, whenever the animal had this direction, the cells would be active. It's active here, it's active here, and it's active here. And it's something so striking that you can actually see it already from the firing of individual neurons. If you look, there is always chains of spikes that symbolize moments in which the animal was moving in a particular direction. If you plot the frequency of firing in relation to the orientation of the head, you have all the 360 degrees, you find that this cell has a very high firing rate when the animal is pointing east, and is almost silent when the animal is pointing in another direction. Another way to look at this is that if you look on a time-to-time basis, so as the animal moves, and you plot the orientation of the head in degrees, you see that the cell is firing always when the animal has an orientation that is around 360/0. These are the equivalent of the compass that we were talking about before. These are what we call now head direction cells. So head direction cells are cells that are fired whenever an animal is facing a specific direction in allocentric coordinate. And allocentric coordinate means that it's based on the reference word of the room. It's always when the animal is facing east or west or north or south, not when the animal is facing on his left or on his right. This is one of the principles of the hippocampus. The hippocampus harbors an allocentric map, and this is what this afternoon Maria is going to talk to you about also when we study how the animal uses these cells or this area of the brain to actually orient itself. The research went on. Now we are more or less in the 80s, beginning of the 90s. John O'Keefe did the first discoveries at the beginning of the 70s. Now we have to do a jump up to the 2000s, so apart like 15 years ago, to have people that start to record from the area of the brain that is known as the medial entorhinal cortex, which, remember, this is the gate or the node that connects information from the rest of the brain toward the hippocampus. People developed and introduced basically tetrodes here, and what they found was actually quite striking. Cells, some of the cells I would say, were firing where the animal was close to one of the walls. And this was actually very plastic. It was the wall. It wasn't a place or a place field, because if you added the new wall in the middle of the room, you could see that another firing specificity would appear. And if you remove that middle wall, then that firing specificity actually is almost completely gone. So these cells were cells that tells the animal there is a border here. There is something that you cannot cross, that you cannot go beyond. The space is done. You cannot walk through it. At least that's our interpretation of it, right? So border cells are cells that fire where the animal is close to a border, again in allocentric coordinates. So either on this border or on this border, but even if they go in this direction, so the border is on their left, or if they go in this direction, so the border is on their right. Importantly, the border cell maintains its firing even in darkness when you cannot see the border, or even if you remove the possibility to touch the border. If you create a virtual reality environment similar to the one that we saw yesterday, but in which you don't have fixed the animal, but you have leave the animal rotates at free will, you still see this type of responses in location where a border is there, meaning that the animal is not able to go further when he moves forward. Is that a clear concept? This is really a message to make you pass that these cells do not depend just on one sensory modality for their activity, but you can actually manipulate the sensory modality that you get. And still these cells are able to rely on other inputs to actually create the representation. All right. Okay, any question about this so far? We're going to go into the last two cells that I want to talk to you about. All right, so fast forward to 2015, we still record from the lateral, the middle and frontal cortex, and we do an experiment that is being dubbed by my previous supervisor as the Flintstones car experiment. So you put rat into a car that they can move when they walk. You remember as the Flintstone. I mean, if you haven't watched the Flintstone, I will feel really old. So please just nod with their head to boost my self-esteem. But the thing is that the animal can move and you can control the speed of the animal when the animal is moving. And what they find is they find cells whose firing rate, depicted here in green, is able to linearly increase as the firing rate of the animal increases, as the running speed, sorry, of the animal increases. These, of course, with not much fantasy, I should say, they've been called the speed cells. And the speed cells are cells that fire with a linear relationship to the movement of the animal, to the speed of the animal. Now, speed is a tricky thing for an animal because you can run faster because you're excited, because you're scared, because you're more attentive, or because you just are basically like, can't wait to arrive to the place where you want to go. So there is a lot of different variables that might influence while the animal is running faster. However, the cell is not responding to alertness or to arousal or to fear or to anxiety, because you can look at the increase in the firing rate and establish a linear relationship. You can manipulate the condition in which the animal runs and still see that the animal is running in that specific, that cell is firing with that specific linear relationship. So these are cells that code purely for speed, on the contrary of many other areas of the brain that are still modulated by speed, but they're not really encoding by speed. And the last cell that I want to introduce you is also the most interesting for us. It came again from the middle and frontal cortex layer 2. And introducing these cells, I again want to play you one of the movies that we saw before. You again will see that there will be a gray line that is a representation of the animal running around. Blue, in this case, black dots that will appear, which represent the moment in which the cell was active. And I want you to pay attention to what happens in this particular area of the environment. So at the beginning, you see that the animal is basically, the cell is more or less active in many locations. It's not a place cell, right? It's not active only at the border, so it's not a border cell. And it seems to be active both in instances in which the animal moves eastward, westward, or whatever. It's not really a head direction cells. So at the beginning, these cells were puzzling because they were really like, not really fitting our idea of the other things that were there. Until people realized by recording in larger and larger environments that yes, these cells were active in many places of the environment, but these places were not random. These places were arranged according to a specific symmetry that defines triangles. You can tile the position in which the cell is firing in a with triangular shape to create an hexagon, which is basically a combination from different triangles. And you would be able to predict where the firing fields or the activity of these neurons would be modulated. These, again, with very little, let's say, fantasy, has been called the grid cells. And it can be considered as a neuron that is active in discrete location of the environment, multiple discrete locations of the environment. These locations are arranged in a periodical triangular array that defines a two-dimensional environment and creates the grid. As for the place cells, it does not matter where the animal is coming from or where it is going. The grid cells will fire in that specific location, whichever is the constellation of sensory stimulus that the animal is immediately experiencing. So again, this is not driven solely by one sensory modality, but integrates multiple sensory modality to understand where the animal is, which is something extremely fascinating. The other point, going back to Fabio's question, is that in this case, if you enlarge if you move a border further and further away, the location of these fields and the size of these fields is not going to change. You will just increase number of firing fields as you go. So it seems that this is a universal representation that you lay on the environment that you actually move in, and that basically adapts to the geometry of the environment in which you move in, but it's not directly defined by the environment in which you move in. So to go back, yeah.
Sorry, another question relating to all those experiments. Are they also done in three-dimensional directions? So to look what happens if, I don't know, have possibility to climb, and what will happen to those cells when they reach other dimensions?
Yeah, so this was another line of research. I'm not going to talk about this right now, but I'm going to be telling you what the answer is. So you can do this experiment in two ways, right? The 3D experiment. One, you can do the experiment on a rat, but the rat does not necessarily fly. So it doesn't really move in 3 dimensions, in the sense that it's always moving on a flat surface that can go up and down, but is always a flat surface, the one that is going to be experiencing. So in the end, if you do this experiment in a rat, you're not be able to, let's say, see if the correlates that you see are actually naturalistic or not, which is all the point that John O'Keefe was trying to make. We should listen to neurons when the animal does something that the animal should be doing, right? So the way in which people have approached this question is to record from a species that it's a rodent but flies. And this is bats. And in this case, when you record from hippocampal cells in a bat that is flying, you see that the place field, that is the activity where the cell is active, it's a ball. So it has definitely included a certain, representation of altitude of where it is, because it doesn't fly if the animal goes too high, it doesn't fly if the animal goes too low. If you look at the grid cells, it also seems to have 3-dimensional firing fields, which are arranged not with the precise triangular symmetry that you see here. The head direction cells, some of them encode for a direction in a plane, some of them at that point encode also for pitch, so if the animal is facing upwards or is facing downwards. So in animals that can fly, this seems to be conserved when we go 3-dimensionally. Incidentally, this type of experiment has been done also in rat, because why not? In rat that climbs, for example, mazes that are 3-dimensional. And in that case, they also see that there is a certain representation of height where the animal is that is encoded in this representation when you go to 3 dimensions. So in the end, it seems that this might be a construct that is used and is optimized for 2 dimensions, because that's where a rat moves, but that can be actually like used also to navigate 3-dimensional spaces. Any other question before we move forward? Okay. So at the end, we wanted 4 different devices, something that tells us where we are and where we want to go, something that tells us what we can get there, something that acts as a compass, and in the end, we find the device that we want. Where do you want to go is the place cells, where are you heading is the head direction cells, where are you and how far it is and in which direction you have to move is the grid cells, and how can you get there without bumping into obstacles, it's the border cells. Of course, these are all simplification, if you want, anthropomorphization of the message that these individual cells give that necessarily come from the fact that we interpret what we see with our own mind. But in theory, it really gives you all the information that you need to have to create a representation of space in your brain, to locate yourself in this representation of space, and also to compute angles and vectors that you need to understand how far you're going, in which direction, and which speed you're moving. Okay? So why now is the grid cell so important for neuroscience? We have seen in V1 that the response of neurons might be correlated with pattern by which sensory receptors in the retina are simulated. You can literally see, and you have seen it with your exercise, that a sensory neuron that is in V1 responds to the fact that there is some new photoreceptor in the retina that are simulated because there is one object in the visual field that is in a specific position. And you can create a relation that is almost one-to-one with the stimulation of sensory receptor in the periphery, that is at the eye, with the response that the cell has in V1. This, of course, is true for many cells, not for all the cells that are in V1. So again, this is amplification if you want to pass a message. The fact is, though, that nothing in the world we move repeats with an hexagonal symmetry like the one that is exhibited by the grid cell. And yet the grid cells knows to fire with an hexagonal pattern. This because these cells are one of the most extraordinary examples, in my opinion, and I'm gonna be very excited about this right now, of how neurons in eye-end cortices do not reflect a simple pattern of activity of the sensory areas, but are able to integrate information coming from different sensory modalities and create completely abstract tuning curves, which are basically produced locally, but the way in which neurons interact with each other. They don't inherit a representation from the outside world, but they are able to create something on their own based on the sensory stimulation that the animal receives. And as that, they've been considered as one of the other cells that are among the few examples of the neural code that is able to be created to build something completely on its own. And as these, they've been used to understand how to create, how to understand how the brain works, how the brain is able to create something starting from sensory stimuli and giving rise to processes, to cognitive processes like navigation, like formation of memory, like the attachment of valence to individual experiences. I'm gonna go now a little bit more on the on a sort of a more, okay, so this is strange, but I'm gonna go a little bit in trying to go a little bit deeper into these concepts, into trying to understand how can a grid cells, a place cells, a direction cells know where to fire, and how local connectivity motifs might actually be important, the one that you studied yesterday, for creating this type of tuning curves that we see in higher sensory areas. This is a little bit of a complex concept that it's hard to grasp, but I'm trying to get it as easy as I can for you to basically have a very strong intuition of how these things will work. So place and head direction cells fighting tuning curves might be generated by the way in which neurons connect to each other between excitatory and inhibitory circuit. And if you remember, this is a combination of the type of connectivity motifs that we had distinguished yesterday, right? What we're gonna do now is to create a model of connectivity or network architecture that might explain the way in which the grid cells and the head direction cells will fire. And in the exercise that we're gonna do as soon as we finish here, we're gonna test a little bit how this might work. So the grid might be considered as a production of a local network computation. Immediately as soon as the grid has been discovered, people have thought that this comes from the way in which neurons are connected and interact with each other. And in particular, they've created a model in the same way that you have created network architectures yesterday that might produce the grid by the way in which activity propagates in the network. And they've called this type of model the attractor network. Now, an attractor network is something quite easy to grasp, but quite fundamental and important if you want to understand how the brain works away from the sensory areas. So this is a network that is organized according to the principle. The principle is that some cells are very strongly connected with each other. Some excitatory neurons are basically making synapses with each other that are very, very strong so that they are able to mutually excite each other when one is active. but they connect very loosely with neurons that are far away into the network. So imagine that you lay down neurons in your Neuronify network to take few neurons in a grid, right? So we have cell number 1, number 2, number 3, number 4, and so on. And as you did yesterday, you create connection between neurons that are close by, very strong, and you create very weak connection with neurons that are very far away. So much so that when you end up in this blue area, these neurons are actually able to inhibit each other's activity through inhibitory neurons. In a network that is created like this, and we're going to create it now as we go into Neuronify, a little bit of simulation create a persistent way, a persistent activity that is called a bump of activity. The persistent activity is the same thing that I was showing you yesterday with this activity that reverberates and stays constantly active in the network, right? Now, if you were able to couple the movement of the bump of activity in the network to the movement of the animal, so that when the animal moves a little bit on the left, also the bump of activity moves a little bit on the left, and so on and so forth, you would be able to create a dynamic pattern of neural activity that follows the animal as it moves into the sensory environment. And by tricking the network and making it look like a torus, like basically a donut, the movement of the animal in space would just create a stereotyped and repeating pattern of activity in the cells of the network that show up as an hexagonal-like grid pattern. The grid cells was discovered in 2004. This particular model was put forward in 2006. And since then, we are trying to look at things that could help us basically verify the model, meaning that to see if the model was actually true, if this is the way in which grid cells are produced in the brain, or to nullify the model. We have identified that indeed connectivity is organized like this and in a rigid way just recently. And we in the lab are working on trying to understand if we find these dynamic patterns of activity that are basically like supporting the movement of the activity bump into the medial and frontal cortex network. Of course, we are not the only one doing it in the world, but we are one of those that are tackling these questions. In the same way that you have created an activity, sorry, a continuous attractor, flat activity, continuous attractor for the grid cells, you can model the head direction cells with a ring attractor, in which, again, neurons, instead of being on a plane, are arranged in a grid, in a ring, and they are connected strongly with each other if they're close by, but they're able to inhibit each other if they're farther away. Again, if you have this type of connectivity, an activity bump emerges here that you would be able to move clockwise or anti-clockwise, depending on the animal movement. And on a single cell level, that will give rise to individual tuning curves that are basically tuned to a specific orientation of the animal in allocentric coordinates, which is the head direction cells. And that the head direction system works like this has actually been shown by few landmark papers, both in the mouse and in the fly. This is also present in the fly in very similar ways, and is called ring attractors. By now, we know that attractor networks are extremely pervasive in the cognitive areas of our brain. They are basically supporting function like episodic memories, so memory of everyday experiences, in point attractor in CA3, and this is what we are going to look at tomorrow. They support working memory with persistent attractors in the medial prefrontal cortex. They support decision-making with point attractors, again, in the medial prefrontal cortex, and they support navigation with ring, point, or continuous attractor in the hippocampus and in the MEC. So attractor network might be the way in which the neural code in high-end cortices is created. So if you look at the graph that I've already showed you yesterday of the cortical hierarchy, at the bottom of which you find the sensory area, and at the top of which you find the association areas that are able to integrate information from multiple sensory modalities, in this area here, you can find a specific connectivity motif and network architecture, which are able to put together information coming from different sensory modalities and build representation that go beyond an individual sensory stimulation, like the concept cells, like the grid cells, like the place cells, like the head direction cells. In these areas, tuning curves might emerge because of the local way in which neurons are connected to each other, and because of the specific network architecture that are built by connecting neurons with each other. This is how and where most likely memory, reasoning, and imagination arises. So this is the last concept. You can imagine the brain as going into different type of computation. an outside-in computation, namely that the computation that are in the tuning curves that are located in this area are mostly driven by what happened in the world outside of you. So in the way in which sensory receptors are stimulated, and something that instead goes in the opposite direction, in which computation are created by the way internally, by the way two neurons are connected to each other. And we will see on Friday how these computations are actually able to always communicate with the other computation and create, again, a balance and an interaction and an interplay between the two. You use the senses to create your memories. You use your memories to guide your action and to actually predict what you expect to see in the sensory world around yourself. This is something we're going to see on Friday. All right. I went a little bit longer than I thought, so we have a little time for questions, but I think this is actually something that if you're interested in, we can definitely discuss more and more later. But if you have any question about these, about the concept of the attractor, or about the idea of neural code in high-end cortices, please let me know now, and we can go through this together.
There's a question at Slido.
Okay, let me check that out. Thank you for reminding. Yeah, so what is the input stimulus needed for the head direction cells to be able to function properly? Inner ear, visual, and so on. So both. So the inner ear is the vestibular system that tells you in which direction you're actually moving. And this is absolutely necessary for the ring attractor to actually come up and to be coordinated. So in a ring attractor, the neuron that, let me just go back to the slide so you maybe can see this better. So the architecture of the ring attractor means that the neuron in red here is always have to fire close to the neuron when the animal is facing in a direction that is close to the one on the neuron orange here. And that is, again, close to the one that fires when the animal is facing in this direction here. So it is important for a certain type of let's say, rigidity in the network. So the relationship of activity between these neurons are very fixed, because otherwise you would never know if you're moving in one direction or in the other. You have to have like a fixed representation in the relationship between the individual neurons. And that seems to be given by the fact that you have a vestibular system that actually drives the building of this network during development in a way that it's very rigid. However, if you don't have visual input because you're blind or because you have like, you still didn't open your eyes in mice, let's say, the relationship between the red one and the orange one always remains constant. So they always are active when the animal faces in direction that are very, very similar. But they're not always at the same position. So basically you have this circle that starts spinning, is not anchored. The north is not always the north. The north might be facing north in one instance, then drift and direct west in another instance, then drift and direct south in another instance, and so on. So you need the inner ear or the vestibular system to actually create coherence between the neurons of the ring, and you need the visual input to actually anchor this representation to the world outside of you. And this is something that is quite general for this type of representation, even for the grid. You need an input to anchor your universal representation, your universal grid to the environment in which you're moving. So you have an internal representation and you use an immediate sensory stimulus to anchor this representation to the map, to really turn the map and adapt it to the area that you're actually moving on. So I hope I satisfied the question of the person that was asking that.
I also have a question regarding the grid cells. Are the grids different from individual to individual, for example, the orientation or size, or is there some sort of hardwiring that says the grid has to be in this and this size?
Yeah, so you're asking between different grids or between different people?
Between different individuals.
Okay, so so this is a great question. This is part of the things that we're actually studying. How much of this network architecture is hardwired during development? And this is something that is still an open question, because during development, when the animal starts moving, you don't see grid cells. And the grid cell starts to appear only as the animal grows older. So you might imagine that you use the first time that you actually explore on your own the world to build a representation of the world that then you will use for the whole experience. So the difference between animal to animal exists. Different grid cells come in different sizes, meaning that you can have, the hexagon. One grid field might be 50 centimeters in one cell, and it might be 1 meter in another cell in the same animal. And this seems not to be random, namely that you have different scales, right? Different magnification. You have the one in which the grid size is 50 centimeters, the other one in which is 1 meter, the other one in which is 3 meters, and so on and so forth. The absolute values of these sizes are different between animal to animal. So the smallest, the most precise one is 50 centimeters in one animal, and it's 70 centimeters in the other animal. However, the ratio between the jumps from 50 centimeters to 1 meter, from 1 meter to 3 meters, and so on, is very stereotyped, even in between people. So if in one animal, the smallest grid is 50 centimeters, and then the next step is 1 meter, in another animal, the first step is going to be 70 centimeters, and the second step is going to be 1 meter and 4. So, the ratio between the two is constant, but the absolute values is actually different. The big question would be, does this difference come from, quirks or different way in which the network is connected, or by the different experience that the individual has perceived or received during the phase in which these grid cells appear? This is something that we are really interested in understanding in the lab, and something for which we're doing some experiments, but for which we have basically yet, well, no evidence is not true. We have little evidence.
Thank you.
Yeah, thanks. One thing that I want to add you though, if you're curious. So I told you that there are grids that are extremely large, in which one grid field is large, like 1.5 meter, right? So you imagine the grid is huge. It's like you cannot visualize it unless you put an animal into a football field, right? To go back to the soccer team. Now you can ask one thing: if the animal never experienced spaces that are that large, because it's always in a small cage and it's never really experiencing such a big field, will that grid cells that is able to take care of telling distances away that are meters and meters apart there, or is it something that is not there because the animal has never experienced that? The interesting thing is that that cell is going to be there. And the first time that you put an animal in a mega space, for example, you put a rat in a 4 to 4 meter box, you will be able to see that size of the grid. It's not that the grid is not there because the animal has never experienced that environment. So some sort of hardwiring already, this experiment tells us that there should be. The amount of that is still unknown. Any other question about this part? Okay, so what we're gonna do now is to take a break. This was a little bit long, so we can take 20 minutes. We see at 2:30 again. And we're gonna look at some complex network architectures and the way in which these neurons are connected. First here, all together, and then you will explore this that you should have on Adam, so you should download them in your Neuronify so that you can test what would happen if you change some of the properties of neuron, add some other neurons, and so on. Just be creative with those type of manipulation. Okay, good. See you in a bit then.